Hello there. Welcome to Fundamentals of Biology, Lesson 5, where we're going to be having a look at nucleic acids. During this session then, I'm going to talk to you a little bit about nucleic acids. So nucleic acids are going to be the third of our 4 classes of organic compounds that we're going to look at. So far we've looked at carbohydrates, and in the last session we looked at lipids. In this session, we're going to look at nucleic acids, and then in the next session, we'll finish off this little section by looking at proteins. So we'll do an introduction to nucleic acids and talk a little bit about what they are and where we find them. And then we'll look at the structure and function of the 2 types of nucleic acid that we get in our in life. And they are DNA and RNA. So we'll talk a bit about the structure and function of each of those. And then as we go through, I'll talk to you about something called the triplet code. Nucleic acids then allow organisms to transfer genetic information from one generation to the next. So you're all familiar with DNA and the fact that DNA exists in our cells in the form of this double helix. And you're probably also familiar with the term genes and genetic. Well, what this basically means is we've got our DNA that's inside the cells of all living organisms, and when organisms reproduce, they pass their DNA, their genes, to the next generation, to their offspring. And what this basically does is it enables them to produce the proteins that they need to build their own bodies and to carry out their own functions. So when we look at our nucleic acids then, we see that there are 2 main types. We get DNA, which its full name is deoxyribonucleic acid. And then we also get RNA, which is ribonucleic acid. And we'll talk a little bit about the differences in their structure as we go through. But this diagram here highlights some of them quite nicely. So on the right hand side here we can see DNA, and it exists it's double stranded, and it exists in this double helix form. And then on the left we can see a strand of RNA. And we can see that it's just a single strand. It doesn't have that opposing strand to make it a double-stranded molecule. These other bits that you can see on the side here, these are what we refer to as nitrogenous bases. And these make up an important part of the monomers that are going to make up our DNA and our RNA. So if you remember from the previous sessions, monomers are the individual building blocks of large macromolecules, and those macromolecules would be referred to as polymers. So RNA and DNA would be polymers, and our monomers that make up DNA and RNA are going to be these monomers called nucleotides, and we'll look at those in a minute. But these nitrogenous bases are an important component of those nucleotides. Okay, so where do we find it then? Well, DNA is organized into chromosomes, and we find it within the nuclei of our cells. And inside the cells, and we'll look at cells in a later lesson, but inside the cells we've got another separate compartment, if you like, and that's called the nucleus. And inside the nucleus you'll find the DNA. And the RNA actually exists in a few different forms, which we'll talk about in a bit. And the RNA can be found inside the nucleus, but it can also be found in the cytoplasm of the cell, which is the bit between the nucleus and the membrane of the cell. And DNA basically contains the instructions for cellular activities. Particularly, it carries the instructions for building proteins. So when organisms produce offspring, those instructions for building proteins are passed down. That's why we often look similar to our parents or maybe to our grandparents, because we're essentially made up of the same proteins. So those instructions for building those proteins is essentially what DNA is. And RNA is essential to the production of those proteins. So the way I always like to think about this is, if you were fortunate enough to win the lottery at the weekend and you wanted to have your dream house built, The first person that you'd probably go to would be an architect. And you'd go to the architect, and then they would they would draw up the blueprint or the plans for your dream house. Now, that blueprint, those plans for building the house, would be our DNA. DNA is just a blueprint. It's just the plans for building our protein or proteins. But our architect then doesn't go out and physically build the building himself or herself. Instead, what they do is they employ builders. And it's the builders that will look at the blueprints for the building. They will look at the plans for the building, and then they will turn that into a physical building. Well, that's RNA. So RNA is the molecules that are actually going to take the instructions or read the instructions for building proteins that are encoded in our DNA, and they're actually going to transfer those instructions into physical proteins that then make up our bodies. So DNA is the plan, RNA is the mechanism by which that plan is turned into physical proteins. So they work very much together in the process of sort of carrying the code, the instructions for building these proteins, and then actually building them. And as we'll see when we go through, and also in the section a lesson that we do on cells, there's a really important organelle that we find within all cells called ribosomes. And ribosomes themselves are made up of a particular type of RNA, and they're like little machines inside our cells that actually do the building. They're the ones that are actually going to physically join together the monomers for proteins to actually build the proteins. So the monomers of proteins are called amino acids. And we'll look at that in more detail in the next section or in the next lesson. But I will mention that a little bit more as we go through, because it's difficult to do a separate lesson for nucleic acids without mentioning proteins. And then likewise, it's difficult to talk about proteins without referring back to DNA and RNA, because they're so heavily interlinked. DNA and RNA basically are responsible for producing our proteins. So I'll talk about proteins and amino acids a fair bit as I go through this. So let's talk about the structure of our nucleic acids then. I'm going to start off by focusing on DNA, and then we'll move on to RNA in a little bit. So cells make nucleic acid polymers then. So this is just a nucleic acid polymer is just another way of saying DNA and RNA. So cells make these nucleic acid polymers by linking together 4 kinds of monomers called nucleotides. So in DNA, the monomers are called nucleotides, and there are 4 different types of nucleotide that we can get. And each of these nucleotides is made up of 3 separate components. 2 of those components will always be the same, but the third component comes in 4 different types. So let's have a look at a simple diagram of a nucleotide. We can see it here. Right at the heart of it, we have a sugar, so a carbohydrate, which we've looked at before. In DNA, this will be the pentose sugar, so 5 carbons, and it will specifically be the pentose sugar called deoxyribose. Hence the name deoxyribonucleic acid for DNA. This sugar here, this carbohydrate, is called deoxyribose. Attached to the deoxyribose, we have this. And we actually saw this. So this is a phosphate group, so it's a phosphorus surrounded by oxygens. And we actually saw these in our last session when we were looking at lipids. We saw these attached to our glycerol molecule along with 2 fatty acids to make a phospholipid. So we see a phosphate group again here in the nucleotides of DNA and RNA. So far we have a pentose sugar, so deoxyribose in DNA. We've got a phosphate group. These 2 bits will always be the same in all of the nucleotides. So all nucleotides will have these 2 bits. Up here, attached to the sugar, we can see a nitrogenous base. And it's these nitrogenous bases that come in 4 different types. And you can see up here, they're often abbreviated to G, A, C, or T. And if you've ever seen any kind of, if you've watched CSI or any kind of crime documentary on TV, and there's sort of genetic analysis involved, you'll often see it represented as a long chain or a long string of letters, G, A, T, T, C, C, A, A, and so on and so forth. Well, what that's referring to is these nitrogenous bases. In this particular example, we can see that this nitrogenous base is adenine. So the A stands for adenine. G, as we'll see in a minute, stands for guanine. C stands for cytosine. And T stands for thymine. So we've got G, A, C, or T. So any one of these four can be attached here. And this itself makes up one single nucleotide. And this one nucleotide is a monomer. It's a monomer, and we are going to join these together over and over and over again to make a single strand of DNA. And then we take a second strand of DNA and we join the two together to make our DNA double helix. So a single chromosome, so I didn't mention this earlier on, but as humans, we have 46 chromosomes, 2 pairs of 23. So we got one set of 23 from our mum, one set of 23 from our dad. Don't go worrying too much about that for now. I'll talk much more about that in a later lesson. So we have 46 chromosomes, and each of our chromosomes is a long strand of DNA, and it will be made up typically of thousands or millions of individual nucleotides joined together. So let's have a slightly closer look at the nitrogenous bases then. We know that they come in 4 different types, so cytosine, guanine, adenine, and thymine. They are split into 2 different types themselves, the purines and the pyrimidines. So the purines, which we can see down here, are adenine and guanine. So they consist of a 6-membered and a 5-membered nitrogen-containing ring fused together. So they're 2 separate rings fused together, and you can see the nitrogen sort of in the structures here, hence the reason they're referred to as a nitrogenous base. The pyrimidines are just single rings, and you can see them up here, and again you can see the nitrogens sort of placed around. And the pyrimidines are made up of thymine and cytosine. Now, don't go worrying too much about this. I think it'll be very rarely in life that you'll ever need to sort of come up with the actual structures or molecular makeup of these. It's just useful to know that they are different in terms of their structure, because as we'll see in a bit, this does affect how they interact with one another. Okay, so just be aware that the nitrogenous bases are 4 different types, so A, T, C, and G, and the 2 different types themselves are the purines and the pyrimidines, and thymine and cytosine are the pyrimidines, and adenine and guanine are the purines. Okay, so how do we go from our nucleotide monomers then, so a single nucleotide which is made up of a pentose sugar, so deoxyribose, attached to a phosphate group and a nitrogenous base, so that's our single monomer. How do we join thousands or millions of those together? We do it through the same process that we've seen so far in the previous 2 sessions. What process do we use to join monomers together to make our larger molecules? I think I can hear you all say it. Yep, it's dehydration synthesis. So we're going to do exactly the same thing here to join our nucleotides together. If we look on the left-hand side of this diagram, we can see 2 nucleotides. They're drawn slightly differently, but let's break it down. So right here in the middle, we can see our pentose sugar. Attached to it here, we can see our nitrogenous base. And then up here, we can see our phosphate group. And then we've got a second nucleotide down here, pentose sugar, nitrogenous base, phosphate group. If we look at the top of this phosphate group, what we can see is that we have there's an oxygen up here, and that can be attached to another another hydrogen. And then if we look at the pentose sugar of this one, we can see a hydroxyl group here, so an oxygen covalently bonded to a hydrogen. So hopefully you know what's going to happen now. An enzyme is needed for this process to occur, but the enzyme will come along, it will attach to these two molecules, and then it will break off two of the hydrogens, one of the oxygens, and that will be given off as H2O, so a water molecule, which if we move to the right over here, we can see this here. There's the water molecule that's given off, and then a new bond is formed between that lone oxygen that's left. So this now joins the pentose sugar of one nucleotide to the phosphate group of another. This is now what we'd refer to as a dinucleotide, so two nucleotides joined together. And this oxygen here represents a link between the two. When we were looking at carbohydrates, the links that were formed by dehydration synthesis were called glycosidic links. In the last session, when we were looking at our triglycerides and we were joining our fatty acids to our glycerol molecules, we made ester links. In this session, when we join two nucleotides together, we make these things, phosphodiester links. Okay, so you do need to remember those three. It's useful to remember them. So glycosidic links in carbohydrates, ester links in our triglycerides, phosphodiester links in nucleotides. And then in the next section, or the next session, when we look at proteins, we'll see that we get a fourth type of link, which are called peptide links that we see in proteins. But for now, when we join two nucleotides together, we make a phosphodiester link. And once we get that, we can start to get a long chain of nucleotides, something that we would call a polynucleotide. Which kind of makes sense, doesn't it? Poly for many, nucleotides. So we're joining many nucleotides together. But what we see when we look at DNA is that it isn't single-stranded. There's two strands together. So if we look at this diagram over here, so let's start with what we would recognize as a DNA double helix. Well, if we were to flatten that out and to straighten it all out, what we would see is something like this. We've just been through where this comes from. So if we look down here at this part of the diagram, what we can see here that I'm highlighting is a pentose sugar of one nucleotide. Up here, that's a phosphate group. Up here, that's a nitrogenous base. Here we can see a phosphodiester link, linking to another nucleotide, linking to another nucleotide, linking to another nucleotide. So what we can see here is four nucleotides, each one joined to the other through a phosphodiester link. And then when we come to get our second strand of DNA, because the two strands are attached to each other via the nitrogenous bases, which you can see here, the opposing strand or the matching strand of DNA has to run in the opposite direction. So the left-hand strand here runs, you could say it runs up with the phosphate group on the left and the nitrogenous base on the right. So it runs in this direction, so from bottom to top. And then the opposing strand has to run in the opposite direction. So the phosphate group on the right and then the nitrogenous base on the left. And that's so these nitrogenous bases can meet each other in the middle. And this is exactly what happens. So when we look at this classic DNA double helix diagram or picture, the ribbons that you see here, the red and the blue handrails of a staircase, if you prefer, these are representing the sugar and the phosphate backbones of the polynucleotides. The stair treads, these little cross braces here that you see, these are representing the nitrogenous bases that basically join the two strands together. And these are joined together, are represented here by these sort of dotted lines. These represent hydrogen bonds. So we get hydrogen bonds holding the two strands together between our nitrogenous bases. So we have the individual strands of DNA, so our polynucleotides, join together by dehydration synthesis, creating these phosphodiester links. The opposing strand of DNA runs in the opposite direction. We actually call that anti-parallel. So they are parallel in the sense that they could go on forever without touching, so they run opposite each other. But anti-parallel means that one strand runs in one direction and the opposing strand runs in the opposite direction. And those two strands that run anti-parallel are joined together by these hydrogen bonds between the nitrogenous bases. As we can see there, then the strands are held together by hydrogen bonds between the pairs of nitrogenous bases. And the next question is really, what is the arrangement of these? Can any of the nitrogenous bases join with any of the others? And the answer to that is no. There is a very specific pattern in which nitrogenous bases can be hydrogen bonded to each other. And it goes back to the structures that we were looking at, the pyrimidines and the purines just now. And what we see is that where we have the base adenine, that will always pair with the base thymine. So adenine and thymine will always pair together, and cytosine and guanine will always pair together. So when you're looking at one strand of DNA, if you have the sequence ATC, you know that the opposing strand will always be TAG. So wherever you see an A, you know that the opposite nucleotide or the opposite nitrogenous base will have to be a T. If you see a T, you know that that will have to match with an A. If you see a C, it has to be paired with a G. And if you see a G, it has to pair with a C. That's really important. That's what we call base pair sequencing. It's A to T, C to G. And this diagram, or these diagrams, show that base pair sequencing in a bit more detail. So we can see up here thymine and adenine forming these two hydrogen bonds between them. This will help to keep those two strands of DNA together. And then down here we can see cytosine and guanine with their three hydrogen bonds between them. And that will help to keep those two strands of DNA together. And here we can see them actually in place. So up here we've got a nucleotide. So this is one strand of DNA. And this is the opposing strand running anti-parallel, so in the opposite direction. And we can see T and A joined together, those two hydrogen bonds holding them together. Here we can see guanine and cytosine, hydrogen bonded together with those three hydrogen bonds. Here's cytosine and guanine again, hydrogen bonded together, and then adenine and thymine at the bottom there. That's basically it for the basic structure of DNA then. Key things to remember are that DNA is a macromolecule. It's a really large molecule. It's a polymer made up of two strands of polynucleotides. So that's a long chain of nucleotides, and we get two of those strands. The nucleotides are joined together by phosphodiester links. And then the two separate strands of DNA, they run anti-parallel, and they're held together by hydrogen bonds. How does DNA code for the production of proteins then? So we've just talked about the fact that DNA is made up of these two strands of polynucleotides. So these two long strands of nucleotides all joined together. It's the sequence of those nucleotides that will actually carry the instructions for the building of proteins. So the order of those nucleotides, so the sequences of the A, T, C's and G's, it's not random. It's that specific sequence of the nucleotides that we are inheriting from our parents and then that we pass on to our own offspring. And it's very, very specific. The specific sequences of nucleotides will relate to specific proteins. And very minor changes in the sequence of these nucleotides will in turn lead to subtle changes in the structure of the proteins that are made. And that itself will then lead to slight changes or sometimes large changes in the characteristics that we see in the organisms when those genes are expressed in the form of proteins. So the way I always tend to think about it really is it's like letters of the alphabet and words. So if you want to put together, if you want to write a word down like elephant, for instance, you need to be able to put the right letters in the right order to spell the word elephant. If you take any other letters from the alphabet and put them together, then you're not going to get elephant. You'll get something different altogether. And so is the relationship between our DNA and our proteins. Everything has to be in the right order to ultimately give us the right protein. And this we'll touch back on this a bit next in the next session when we talk about proteins. But first, let me talk to you a little bit about the way that these nucleotides are structured and how they lead to proteins then. And it's something called the triplet code. The DNA triplet code is the sequence of nucleotides in DNA and RNA that determines the amino acid sequence of proteins. So proteins are polymers. They're really large molecules made up of a long chain of monomers. And then, as we'll see in the next session, they're then highly folded. But at their heart, they're just a long chain of amino acids. And there are 20 different amino acids that make up our proteins. And the order or the sequence of the amino acids will ultimately determine what our protein becomes. And our DNA is what determines the sequence that those amino acids will go into for our proteins. And that is what the triplet code does. The triplet code is the sequence of nucleotides that will determine the sequence of amino acids that makes up our proteins. And we can break this down into things called codons. So when we look at a section of DNA, so a particular section of DNA that carries all of the instructions for building a single or particular protein is what we would refer to as a gene. So that is what a gene is. It's a section of DNA that carries all of the instructions for building a particular protein. So when we look at that protein, and let's say, for instance, it's made up of 1,000 nucleotides, that the specific sequence of nucleotides will be what determines the sequence of amino acids that go into our protein. And the way it works is that out of those 1,000 nucleotides, each 3 adjacent nucleotides make up something called a codon. So 3 adjacent nucleotides constitute a codon, and each codon codes for a single amino acid. So if we look at the diagram over here, we can see a section of RNA. So we'll look at RNA in a moment, but RNA is copied from a section of DNA. And here we can see 3 adjacent nucleotides. Hopefully you've noticed that there's a U in there, and I haven't mentioned anything about U yet, but I will in a minute. But these are just 3 nucleotides. This would make up a codon. And these 3 specific nucleotides in this arrangement, so A, U, and G, will represent a particular amino acid. So this will tell the machinery of the cell, particularly that organelle called the ribosome, that when it's making its protein, it needs to put the amino acid that is coded for by this sequence of nucleotides, it needs to put that one first. And then it will read the next 3 nucleotides. So in this case, we've got A, C, and G. Well, A, C, and G will relate to a particular amino acid. So the ribosome will read this, and it will then know that it needs to get the particular amino acid that this codes for, and it needs to put it second. Then it will move along, and it will read GAG, and GAG will relate to a particular amino acid, and the ribosome will then put that amino acid third, and so on and so forth. The ribosome will go along, and it will read each codon in turn, and it will take the associated amino acid and it will put it into the specific place that it's meant to be. And that means that when we're building our proteins, we will end up with a long chain of amino acids all in the correct order. And as we'll see in the next session on proteins, it's the specific sequence of amino acids that will ultimately give us the protein. So that's going to be really important. What we tend to find, and you might, if you're particularly good at maths, you would have worked this out already. But what we typically find is, and I mentioned just now that there are 20 amino acids that make up our proteins. But when we look at the fact that we have 4 different nucleotides, you can arrange those into 64 different codons. So we've got 64 different codons that actually code for our 20 amino acids. This is ultimately, it means that we have different codons, multiple codons that can code for the same amino acid. And this is a code that we call degenerate because we have multiple different codons that code for the same amino acid. And this just comes down to simple maths. If you were, so when evolution was sort of working on this process, if you were looking at making a codon 2 adjacent nucleotides, you wouldn't have got enough different permutations out to cover the 20 amino acids. So the next step is to go up to 3 adjacent nucleotides. But 3 adjacent nucleotides, when you have 4 possible different nucleotides, well, suddenly you get a large step up, and now we have 64 possible codons. So you have way more than you need, but it's the smallest number that you can get to while still covering the 20 amino acids. So we end up with a lot of surplus, but it's still the most efficient that we could get to. So we have 61 codons that code for the 20 amino acids. And then there are 3 codons that are stop codons that actually stop the process of copying and making our proteins. If we go to the next slide, then I don't want you to worry too much about this, but this is just a table that shows us which of our 20 amino acids are coded for by our codons. So if we take a sequence of nucleotides in our DNA, and say we've got the first nucleotide is T, and then the second letter is C, so thymine, cytosine, and then let's say that the third one is A. So what we can do is we can go T, C, A, and then we can move along, and then what we can see is that that particular codon will code for the amino acid serine. If we go C, A, T, so C, A, T, that gives us histidine. So what you can see there is how each of our codons, our 3 adjacent nucleotides, will relate to a specific amino acid. And this tells the cell then, by reading the codons within our DNA and RNA, it tells the machinery of the cells which amino acids to put in which order when it's building its protein. It's quite a nice little unique system. So that covers us for a very brief and quite quick introduction to DNA then. And like I said, we're going to come back to that in a bit more next session when we look at proteins, but we'll also look at it in a bit more detail in a later lesson when we look at cells, and also when we look at cell division, and then also again when we do a little introduction to genetics. Although this is quite a quick introduction to DNA, this is just to really introduce you to the basics of the topic, so it will set the seed or set the foundation for when we go into it in a bit more detail in later lessons. I next want to just cover RNA in a little bit and talk to you about some of the key differences, structural differences between DNA and RNA. If you remember what I said at the beginning of this, DNA it carries the plan, it carries the instructions for building our proteins. RNA is actually going to do the heavy lifting, if you like. RNA is going to turn those instructions into actual proteins. And I'll talk to you a little bit about that on the next slide, but first let's have a look at the structural differences between RNA and DNA. So at first glance, RNA looks quite a lot like DNA. It's still made up itself of nucleotides. What we can see here, we just take this this second one down, we can see that it's a nucleotide. So we've got a pentose sugar, we've got a nitrogenous base, and we've got a phosphate group. The nucleotides attach to each other from the pentose sugar of one to the phosphate group of another through a phosphodiester link. So it's still all the same basic principles that we looked at with DNA. But some structural differences then are that RNA has a different sugar. So this pentose sugar, this 5-carbon sugar, is called ribose instead of deoxyribose. The next thing that we notice, and I touched on this on one of the previous slides, is that one of the nitrogenous bases is different. So in DNA, we had adenine, thymine, cytosine, and guanine, and our base pair sequencing was A to T, C to G. Well, when we look at RNA, we see that we have these four. We don't have thymine anymore. Instead of thymine, we have a base called uracil. And we see that the base pair sequencing is still the same though. Adenine now, instead of bonding to thymine, will bond to uracil. So it's A to U. And then guanine and cytosine will still go together. So it's A to U and C to G when we're dealing with RNA. It's not anything that you need to worry about too much. Just remember that in RNA, you will see U's instead of T's. But they do essentially the same job. What we also see with RNA is typically it's much shorter than a section of DNA. So DNA that makes up a single chromosome will carry dozens or hundreds and sometimes thousands of individual genes, whereas RNA is just copied from a short section of DNA, typically to cover a single gene. So it tends to be a lot shorter than DNA. And what we also find is that RNA comes in a lot of different types. So there are dozens of different types of RNA in the natural world. But when it comes to our own cells, and when we're looking at the transfer of information from DNA to the building of proteins, we see that there are 3 main types. So RNA comes in 3 main types, and we'll look at that on the next slide. Okay, so here are our 3 main types of RNA then. And first down here we can see I've written the sequence that the process goes from the instructions in DNA, and then ultimately we end up with our physical protein being built. And then we can see that we have 3 different types of RNA linking the two different points. So we've got the written instructions in the form of DNA, so our sequence of nucleotides, and then we ultimately end up with a protein. And what we can see first is we have this thing called mRNA, which stands for messenger RNA. And messenger RNA is a short section of RNA that is copied from our DNA. So we have a special enzyme called RNA polymerase that will zip along the gene, so zip along the section of DNA, and as it reads the sequence of nucleotides, it will start producing a matching or corresponding section of messenger RNA. And here you can see the different color-coded nitrogenous bases here. So each one of the nucleotides that make up this section of messenger RNA is the perfect matching strand for that section of DNA that it's been copied from. The messenger RNA, then it goes through a period of processing and sort of chopping and changing, but we don't need to worry about that for now. The messenger RNA then will then move out of the nucleus of the cell. It will move into what's called the cytoplasm, where it will dock, if you like, or sort of join up with the organelle called the ribosome that we mentioned earlier on. And ribosomes are the site of protein synthesis. They're little machines that build our proteins. And ribosomes themselves are made up of a type of RNA. They're actually made up of a mixture of RNA and protein. And the type of RNA that makes them up is called rRNA, or ribosomal RNA. And what the ribosomal RNA and the mixed with the protein makes up is these ribosomes. And what they do is they will read the sequence of nucleotides that make up our section of messenger RNA. So you can imagine that this will fit in between these two different subunits, and it zips through a bit like a barcode reader. And as the ribosome reads a single codon, so 3 adjacent nucleotides, it knows that corresponds to a particular amino acid that we looked at on the table just now. And what the ribosome will then do is it will send out for that particular amino acid to be brought in to the ribosome. And the way that those amino acids are brought in to the ribosome is where we see our third type of RNA, and it's here. It's called tRNA, or transfer RNA. So we can see our section of transfer RNA here, and it's carrying our amino acid. So the transfer RNA will deliver the amino acids to the ribosomes, where they get joined together through dehydration synthesis to ultimately make our protein. And that's a process, it's a pretty complicated process called protein synthesis. And I will mention it in a bit more detail in a later lesson, but if you wanted to get a head start on that, it's quite an easy one to look up in a good textbook or online. And it's worth having a look at because it is a pretty interesting process. But basically, we can see then that we start off with the instructions for building our protein in the form of the triplet code, the section of the sequence of nucleotides in our DNA. That is copied into a strand of messenger RNA. The messenger RNA moves into the cytoplasm of the cell, where it combines with our ribosomal RNA that makes up our ribosomes. The ribosomes will then take in amino acids delivered to them by the transfer RNA, and they join the amino acids together, ultimately making a protein. Okay, so that's basically RNA. It's a lot like DNA, although if we go back, we can see that it's single-stranded typically. It has a different sugar, so the sugar is called ribose instead of deoxyribose. One of the nucleotides has a different nitrogenous base, so we have uracil instead of thymine. And RNA is typically shorter than DNA because it's copied from a section of DNA. And RNA comes in 3 different main types: messenger RNA, ribosomal RNA, and transfer RNA. DNA is the instructions for building proteins. RNA actually does the building of the protein. Okay, so let's go through 4 quick recap questions then. So I'll read these out and then we can pause the video and then you can have a go at answering them, and then we'll go through the answers together. So question number 1, name the 4 bases found in DNA. Number 2, what replaces one of the bases in RNA? In DNA, describe the base pairing arrangement. And then number 4, what are codons? A little bit hard on that one. So we can pause this now and we'll give you a few minutes to try and answer those questions. So you can just jot them down and then we'll go through the answers together. Okay then, welcome back. Hopefully you had a chance to answer those and there weren't too many troubles. So let's go through the answers together then. So number 1, name the 4 bases found in DNA. Well, hopefully you had adenine, thymine, guanine, and cytosine. So A, T, G, and C. Number 2, what replaces one of the bases in RNA? Well, hopefully you remembered that uracil replaces thymine in RNA. So U replaces T. And then in number 3, in DNA, describe the base pairing arrangement. And that is A to T and C to G. And then number 4, what are codons? So a little bit more difficult to answer this one, but hopefully you had a go at it. So what are codons then? Well, codons are made up of 3 adjacent nucleotides that code for the placement of specific amino acids in a protein. And this makes up the triplet code. Now, likely you worded it slightly differently to that, but as long as you came up with something similar, so codons are part of the triplet code. They are made up of 3 adjacent nucleotides in DNA and RNA, and a single codon codes for the placement of a particular amino acid in a protein. Okay, so that brings us to the end of this session then, where we have talked briefly about nucleic acids, so what they are and what they do and why they're important. And then we've talked a little bit about the structure and function of DNA and also of RNA. And then as we've gone through as well, we've talked a little bit about the triplet code. Now, some of this is a little bit complicated, but don't worry, we will revisit this, or you can revisit it before the next session as well if you want to go back over this and go back over some notes. That will definitely help. But we will touch on some of this again in the next session where we look at proteins, because nucleic acids and proteins are so intrinsically linked. And then, like I said, we'll also talk about DNA in particular in other sessions where we'd start looking at cells, particularly with cell division, and also when we talk a little bit about genetics. But hopefully that was useful, and I'll see you in the next session. Thanks very much.